which is that Google Video, when I tried to load the last class lecture, sent me this little notice saying that you can only upload videos to Google for a little while longer because pretty soon we're going to not let anybody do it anymore. But you can still do it at YouTube. So I went over to look at YouTube, and YouTube has a 10-minute time limit. So I hit up my contacts at Google and sweet-talked them, and they sweet-talked someone at YouTube, and I now have a 50-minute time limit at YouTube. So at some point, there will be a switch from Google Video to YouTube Video, and it'll just be a link, so it shouldn't make a big difference to you. But it hopefully will still work, if not exactly the same way. But my contact at Google is my daughter. So she's a real contact. She's not a fake contact. So the video should still work. There'll probably be some kind of transitional thing, but hopefully they will be okay. Do people have any questions about Wednesday's opening discussion of enzyme kinetics and how they work? Just one more reminder that if you would like to work together with us on study strategies, we warmly welcome you to do that. Changing study strategies now for the rest of the semester is usually effective. The longer people wait to change what they're doing, the harder it is to catch up. So I do encourage people who would like to do better to come and strategize away, and we can give you tools to work with. All right. I'm going to just sort of again repeat the things I said here because I think it's worth making sure they're all current in people's minds before we get into the more complicated, less obvious ones. So we are talking about competitors and how competitors can alter the apparent kinetic properties of enzymes. And one common form of inhibition is competitive inhibition, which is reversible. If it isn't reversible, it's not competitive inhibition. So it's got to be reversible to be competitive inhibition. And competitive inhibition happens when a molecule, the inhibitor, has structural similarities that allow it to bind at the active site. So competitive inhibitors are, by definition, binding at the active site. They'll bind there, but they don't react. And so they dilute out the universe of reactive molecules with these things that are tying up some fraction of the enzymes reversibly in an unproductive way. And the effect of that is this, is that if you look at the kinetics of the enzyme in the absence of the inhibitor, it will have some kind of a Vmax showing the rate at which the reaction goes forward when the enzyme is occupied. And it will have some kind of Km, some concentration of substrate that half saturates the enzyme. In the presence of a competitive inhibitor, some of the time the enzyme is going to be occupied with inhibitor instead of substrate, and that's not productive. And the more and more substrate you add, the smaller and smaller the fraction of time is that it's occupied by the inhibitor, so the more nearly it comes to its normal behavior. And that's what we see in this graph, that if you have enough substrate around, it overwhelms the inhibitor and does not affect the Vmax at the limit. On the other hand, if the inhibitor is around, it takes more substrate to get to the point that half the time the enzyme is occupied, in fact, by substrate rather than inhibitor. So it has an apparent effect on the Km. It doesn't structurally change the enzyme, but the presence of the inhibitor is detectable by a change in the Km, the concentration of substrate it takes to get to the half maximal velocity. It isn't actually changing the affinity for substrate. It's competing for the site, and some of the time that site is occupied with something non-reactive. Okay, any questions about this before we move on to the more Byzantine kinds of inhibitors? Okay. We discussed briefly how using a double reciprocal plot allowed us to detect the presence of a competitive inhibitor, that the green line here, this is 1 over the initial velocity, this is 1 over the substrate, that the vertical axis, the y-axis, gives you 1 over the maximal velocity, and the intercept with the x gives you minus 1 over the Km, the Michaelis constant, which is a measure of affinity, the tendency to hold on to the substrate. And that, consistent with what we just said from the hyperbolic graph, the competitive inhibitor changes the apparent Km by this alpha, the inhibitory constant, making it take more substrate closer to the axis is higher concentration because this is a reciprocal plot, but no effect on the apparent Vmax. So when you look at a Lineweaver-Burk graph, you can detect the kinetic effect of the competitor. So this brings us to an aspect of enzymes that is important to keep in mind, which is that unless the reaction that an enzyme catalyzes is a completely irreversible reaction, a few are, but not very many, that there can be product inhibition. That as A goes to B, if B starts to build up, the B is going to have a tendency to go back to A. It's going to inhibit the forward reaction. And it's the fact of product inhibition that makes it important to measure enzyme kinetic properties at that early stage when you mix the reactants right together and you look at it right then because you haven't made any product yet or made very little product yet, so you're not going to see the effect of product inhibition. So product inhibition is one of the reasons why we always talk about V sub 0, the initial velocity, because that's the velocity before we accumulate enough product to have product inhibition from it. Another thing that we sort of get a glimpse of through thinking about competitive inhibitors is transition state analogs and why transition state analogs are such wonderful competitive inhibitors. So this is the enzyme that we talked about before, triose phosphate isomerase. So here's glyceraldehyde 3-phosphate. It's a triose, a 3-carbon sugar with a phosphate, triose phosphate. Here's dihydroxyacetone phosphate, again another triose. The isomerase has just changed this one into that one, no addition or subtraction of atoms, so it's an isomer. And it had a transition state intermediate like this. This is the ene diolate. So there's the ene group. It was a diol 8 because it's in the ionized state there. So the ene diolate transition state is not something that would form very easily by itself. That's what the enzyme is doing. It's making it, it's lowering it's helping to stabilize that transition state, and that's part of how it's lowering the delta G for the reaction. Well, if we look at this compound, phosphoglycohydroxamate, this is a transition state analog. You can see how the structure resembles the structure of the ene diolate. And because it's a transition state analog, not an analog of the substrate of the product, but of the transition state, it's an especially potent competitive inhibitor. Because that's one of the jobs of this enzyme is to stabilize the transition state. And we can get a little bit of a sense of just how viciously good a competitive inhibitor, a transition analog, can be if we look at this example. This is a different reaction we're going to look at somewhat later in the semester. This is taking adenosine to inosine, and inosine is one of the intermediates along the way to something else. And the Ki, the inhibitory constant of inosine on this reaction, on its own production, is moderate. It's 10 to the minus 4, 3 times 10 to the minus 4. This is the transition state, the compound, the intermediate compound that the enzyme helps to stabilize as its way of catalyzing things. And here is a transition state analog. You can see here how it has the NOH sort of structure there mimicked by here OH just with an H there. And this is an analog of that transition state. And look at its inhibitory constant, 10 to the minus 13th. So it's 10 to the 9th times better at inhibiting this reaction than the product is. That's a very powerful inhibitor. Okay. Now you can get a number of other kinds of inhibition as well. And I want to talk about a few of those as well. Partly because it's important to realize that there's more to it than just competitive inhibition, partly because some of these things turn out to be analogs for thinking about regulation of metabolism. Now let's say we have some enzyme here, and now instead of just Ms. Pac-Man, we've got kind of a punctured washer or something. So this enzyme can do a couple of different things. This enzyme can bind substrate, in which case it's an ES enzyme substrate complex. this empty enzyme can bind an inhibitor. This is not a competitive inhibitor. This is a different kind of inhibitor, but it's still red, sorry. So on the back end side here, this is the inhibitor molecule binding somewhere else, binding other than the active site. And either one of, the enzyme substrate complex can itself bind the inhibitor at that other site that's not the active site. So we have 4 possible states that the enzyme can be in, with just substrate, with just inhibitor, or with the substrate and inhibitor together. And the consequences of this kind of inhibition look like this kinetically. If we look at the original curve of the kinetics, with its asymptotically approaching Vmax, it has a Km that indicates the concentration of substrate that it takes to half saturate it. So this is its measure of affinity. This is how fast it turns over substrate when it's bound. And this particular kind of inhibition that's binding elsewhere on the site is affecting both the Vmax. So you can see that no matter how much substrate we put in, it cannot go full tilt. But it also takes more substrate to get there. So this inhibitor has affected both the affinity for it, how well the enzyme binds the substrate, and the catalytic efficiency of it, the catalytic ability of it, the ability to go forward in the forward reaction. So it's affected both Vmax and Km. So if you just to step back a second and look at the competitive inhibitor and the mixed inhibitor, this should bring home to you that Km and Vmax are independent properties of enzymes. They are different, separate, distinct properties. Vmax, the ability to catalyze the reaction in the forward direction. Km, the affinity for the substrate. And the difference between the competitive inhibitor and the mixed inhibitor really brings that home, because the competitive inhibitor only shows up as a change in the apparent Km, whereas the mixed inhibitor can affect both the Km and the Vmax. When it binds here on the back side of the enzyme, it's like it's tweaking the active site both, so it's not as good at binding the substrate, and it's not as good at catalyzing a reaction. Any questions about this before we go on? There are other kinds of inhibitors that are called non-competitive that can change only the Vmax. They do not affect its ability to bind the substrate. They only affect its ability to catalyze the reaction in the forward direction. So these equations are meant to bring home some of the ways in which inhibitors can bind and how we look at the kinetic constants as a consequence. Here we have free enzyme and free substrate. In our initial discussions of enzyme kinetics, we talked about enzyme and substrate making the enzyme-substrate complex, and then the enzyme-substrate complex being able to go forward and make free enzyme and free product. So there's a forward rate at which it forms enzyme-substrate and a back reaction which it goes to the separate components. Enzyme-substrate complex has a forward rate reaction. There are additional rate constants in the presence of inhibitors. You can talk about the enzyme itself binding the inhibitor. This is Ki. This is an inhibitory constant of just the forward and the back reactions of binding inhibitor, in which case you have an enzyme inhibitor complex. You can also look at the enzyme substrate complex binding inhibitor and making an enzyme substrate inhibitor complex. This is a separate inhibitory constant. So you can start to sort of dissect the possible times and places that the inhibitors can interact with these things. And in this table, it allows us to sort out the consequences of whether the thing binds only to the free enzyme, only binds to the enzyme substrate complex, can bind to either one of them equally well or not. If there's only a Ki, if it only binds to an empty enzyme, if the only question is whether you're going to have an ES complex or an EI complex, then that's what we were talking about as competitive inhibitors. It affects the apparent Km, by modifying it with it reduces the apparent Km, or changes the Km by a factor. But the Vmax is unaffected. We talked about the mixed inhibitors, where there was an effect where there are possibilities of binding to either the empty enzyme or to the enzyme substrate complex, and they're not the same. And in this case, you have both a change in the Km and in the Vmax. In the case of the non-competitives, the tendency to bind to those things is not affected by whether the enzyme's occupied or not. You only see a change in the Vmax. You don't see a change in the other. So this, again, brings home, I hope, that the Vmax, the ability to catalyze that forward reaction, is one property, and the Km, the tendency to bind the substrate, is a separate, distinct property. This comes out, I think, when you look at some of the graphs for these things. And so you can see here, actually, maybe this is a good time to take a small break. Eli and AJ, could you help me out? We're going to take a little break. And I want you to share with us and your fellow students study strategies that have worked well for you. So, Eli and I are having conversations with various students about the study strategies that they might want to work on. And we're interested in hearing from you what 1 or 2 or 3 study strategies that you think have been most important so far in the course. And if you'd like us to know that you're here, you're invited to print your name on it. I will take these cards and I'll compile sort of the best hits list and share that with your classmates anonymously, as I shared the study questions as well. So, and while you're thinking through what study strategies you want to put down, you can also be looking at these graphs and taking a little look at them. So feel free to share whatever study strategies work for you. There may be some novel ones that I don't know about, and I'd love to hear about them. You may have found that, using a whiteboard for structures has been key. You may find that you've got to have, Axl Rose on in the background. Whatever you think has been really, really the most helpful for you. Maybe Bach cantatas do it for you. Do we have enough cards? Cool. Anybody still need a card? Okay. I hope so. Find out sort of tap the dimensions of people's ingenuity. Do people need more time to write? Okay, if you need a little more time, feel free to write, and we can start walking through some of these graphs while you're writing. So these are 3 different figures, all of them looking at how the effects of inhibitors show up using the Lineweaver-Burke or reciprocal plot. These are, again, graphically vivid ways of doing it. If you're going to get into enzyme kinetics in a really serious way, there is now high-powered software on computers that allows you to do it from the actual data directly and not have to do this kind of extrapolation. But these are visual. These, I think, help kind of bring home some of the differences. So on each of these graphs, the concentration of the inhibitor is rising upward like that. And we had talked about competitive inhibitors, and this is showing you that if you start with your uninhibited enzyme and add more and yet more, you get what we talked about. Here's your vertical axis. This is 1 over the Vmax, no change in that Vmax. And this is the 1 over S, so minus 1 over the Km is off there somewhere, over around here maybe. And as you add more and more inhibitor, it takes more and more substrate to compete for that thing. And here's the pure non-competitive case. So here, the minus 1 over S is not changed, so there's no apparent change in the affinity for the substrate. But as you add more and more inhibitor, the Vmax comes down and comes down. So this is only affecting the ability of that enzyme to catalyze the forward reaction. But it binds the substrate just the same. And this is showing you an example of a mixed competitor, the one where we showed like the back-end binding that was going to alter the active site, so it both wasn't as good at binding and it wasn't as good at catalyzing. And it shows up, it's raising 1 over V, so it's making it slower when it's occupied, and it's changing the Km. It takes more substrate to saturate it. Okay? Any other questions about these graphs? Okay. Well, one reason for thinking about inhibitors is because in the course of metabolism, we're going to see that there's a lot of interaction among metabolites and enzymes, and that there's a lot of feedback going on so that the small molecules of metabolism can regulate these great big hulking enzymes. And one of the very common ways that we see that happening is in a phenomenon called allosteric inhibition. And this is basically the idea that an enzyme activity can be controlled by other metabolites. We talked about product inhibition already. That is, that if you have A going to B, as B accumulates, it'll inhibit the forward reaction just by mass action, as long as it's a reversible enzyme. But when we talk about allosteric inhibition, we're talking about something a little bit different. We're talking about the ability of metabolites to bind elsewhere, allosteric, somewhere else on the enzyme and change its behavior. And this particular example that we're going to talk about is an extremely important one. This is an example from glycolysis, so it's worth learning now because you're going to learn it later. Or you can wait, either one. And this is looking at taking fructose 6-phosphate. So here's fructose with a phosphate on the 6th position. And it's going to use an ATP as a phosphate donor, phosphofructokinase. So it's a kinase. It's going to add a phosphate to phosphofructose. And its product is going to be fructose 1,6-bisphosphate. So this is now fructose with a phosphate flapping off each end. And that releases a DP as the other product. And then after a number of other steps, we end up with a 3-carbon fragment, phosphoenol pyruvate. And phosphoenol pyruvate will turn out to be a very important little player in a number of different pathways. But certainly one of the things that phosphofructokinase does is to start sending 6-carbon sugars down a pathway that ends up down around phosphoenol pyruvate. And so what we find is that phosphoenol pyruvate, even though it's several steps removed, can come back and feed back on the reaction to inhibit the activity of phosphofructokinase. So this is an example of feedback inhibition where if you start to accumulate the product, it doesn't make sense to keep doing more of it. You want to shut down that and maybe send your phosphofructose somewhere else and do something else with it. And it inhibits it in an interesting way. So here we have a picture of phosphofructokinase. This is a bacterial form of it. And here we see the kinetics of the phosphofructokinase in the absence of phosphoenolpyruvate. It's a typical hyperbolic behavior. And here we see the kinetics in the presence of phosphoenolpyruvate. And it now has sigmoidal kinetics with respect to phosphofructose, fructose 6-phosphate. So in the presence of a fixed amount of phosphoenolpyruvate, the enzyme now has sigmoidal response to the concentration of fructose 6-phosphate. Its apparent Vmax looks unaffected, but both the shape of the curve and the Km have been altered. And so in the presence of phosphoenolpyruvate, it's going to take quite a lot more fructose 6-phosphate to bind to the enzyme before the reaction goes forward. And this is another example of what we saw with hemoglobin. We talked about a tense and a relaxed state of hemoglobin, that deoxyhemoglobin and oxyhemoglobin were structurally a bit different, so that they had different binding properties. Phosphoenolpyruvate switches the phosphofructokinase into a tense state that is less friendly to binding the fructose 6-phosphate. And interestingly, ADP, and ADP is one of the products of the reaction, ADP helps to keep this in the relaxed state, tends to push it back towards the hyperbolic curve. Now, you might think ADP is a product of it. Why isn't it inhibiting it? Well, this enzyme is part of a key energy-producing pathway, and ADP is a less energetic form of an adenine nucleotide. And so ADP, if it's accumulating, is a sign to the cell that maybe we do need to make more of this. And so the phosphoenolpyruvate and the ADP are signals of opposite things, and the two of them filter in to feed information to the phosphofructokinase. And it's like the phosphofructokinase is a little computer adding and subtracting and multiplying in its head and saying, okay, given this mixture of conditions, the best thing for me to do at this point is X. So they're a very smart little molecule. As we go further into metabolism, we're going to discover that there are really a large number of ways in which we can regulate enzyme activity. We've talked so far about regulation through small molecules binding, but there are actually a large number of ways. We can change the rate at which we make it or at which we degrade it. Protein degradation is every bit as important as protein synthesis, because otherwise we would be all eyeball deep in proteins. And that's here is showing you the example of this, of the synthesis of a new enzyme. We can change location. This is a little diagrammatically here, that by moving an enzyme to a location where the substrate is, you can change the activity. There are ionic signals that can alter enzyme activity. Some enzymes are sensitive to pH changes. Some enzymes are sensitive to calcium concentrations, so that cells may trigger a sudden flux in calcium to turn on an enzyme or something. There are covalent modifications that can be very important, and a couple that we'll see are phosphorylation, where instead of using ATP as a substrate, we're using ATP to add a phosphate group to an enzyme that then changes its activity for a period of time. There's always phosphatases to take that off. There also are sometimes modifications with lipid binding. So there's a number of cute tricks that the cells have to modify enzyme activity in ways that are always relevant to what it is that that enzyme is for. So let's think a little bit about some of these things. Which do you think, right, if you have a mutation at the catalytic residues, so an example of catalytic residues would be the As-Pis-Cir on chymotrypsin. There are catalytic residues on any enzyme. If you make a mutation at the catalytic residues, which do you think is most likely to change, the Km or the Vmax? Someone here said Vmax. Would you like to explain? So in other words, if you're affecting the catalytic residues, you're probably going to alter its ability to do the particular thing that enzyme does to catalyze that reaction, stabilize transition states, whatever. Suppose you have mutations at the specificity site. Remember that pocket in chymotrypsin and trypsin and elastase? What about mutations at the specificity site? Are they more likely to affect Km or Vmax? Don't be shy. Sorry? Okay. You want to take a stab at why they would affect Km? Right, right. Km is mostly a measure of affinity, and the specificity pocket plays a key role in selecting a particular subset. Yeah. Okay. As we've gotten to know more about enzymes, as we've gotten to know more about diseases, we've gotten more cagey about developing inhibitors that are specific for specific diseases and not just kind of like, a shotgun approach to things. And one of the places where there have been remarkable progress is in the field of AIDS and HIV. So AIDS is acquired immunodeficiency syndrome. HIV is human immunodeficiency virus. And AIDS is a consequence of having so much HIV in you that you're starting to wipe out your immune cells. HIV is a single-stranded RNA virus. So there's a single strand of RNA, ribonucleic acid, packed inside the virus. And this is showing you an electron micrograph of it. Here's the RNA in here. And it's packed around with a capsule that has enzymes, it has some proteins, and then there's this coat around it of proteins and lipids that protects that kind of payload while it gets to where it's going. So you've got the RNA, you've got some enzymes, you've got some coat proteins. RNA by itself, if you have a little bit of RNA, maybe you can make a little bit of protein. That's kind of a dead end. And so HIV has a rather sly strategy. It's going to make DNA out of RNA. And this is where we first met this enzyme, reverse transcriptase. So transcription is making RNA from DNA. Reverse transcriptase is making DNA from RNA. And the enzyme actually is rather cute. It actually has two active sites. And so it has one active site that can take an RNA molecule and synthesize a complementary DNA molecule from it. And then it has a second active site that has an RNase activity, a ribonuclease activity, that degrades the RNA and then can use that DNA strand for a second round of synthesis. That DNA synthesizing activity is a little bit promiscuous. It's not too concerned about whether it's an RNA or DNA template. And now we have a double-stranded DNA molecule. So we've gone from having a single strand of RNA to double strand of DNA. Reverse transcriptase in the form we find in HIV is a viral protein. It turns out that we do have for our telomeres an enzyme that has some of the same activities, but it's a different protein. So if we target reverse transcriptase, we should be targeting principally the HIV virus, the HIV. And so this was the logic behind reverse transcriptase inhibitors. And the reverse transcriptase inhibitors follow a strategy that has often been a fruitful strategy. They are analogs of substrates. That is, they kind of look like a substrate chemically. They look enough like a substrate that they can actually be incorporated into the growing chain. But they won't provide a place to continue it. So they can be added into the chain, and then they stop further elongation. They're terminators. They end the elongation of it. So substrate analogs like that act something like the dideoxy NTPs that we use for DNA sequencing. There are some other reverse transcriptase inhibitors that are not analogs, but they simply inhibit the activity of the enzyme. Here's an example of one of the terminators. You can see here it's a 2', 3' dideoxy. Remember the dideoxys we used for sequencing? Here's a dideoxy that is powerful because it's going to be added and then not go anywhere. So that's one point of attack that we had for HIV, was hitting the reverse transcriptase. But the life history of HIV gives us other places to attack, and they have also been fruitful. It turns out that small organisms, like viruses and like some bacteria, get more bang for their buck by synthesizing an RNA molecule that then is translated into a big piece of protein that's then cut up into smaller proteins. And this is called a polyprotein. So you make one RNA molecule, you translate one big piece of protein, and then cut it to get several from that one. And this is showing you how the structure of the HIV genome. Each of these things that's a whole block is a polyprotein, and the dotted lines are showing you how many pieces it gets cut up into. So this is a structural protein here, polyprotein. This is a structural polyprotein that's going to make 2. This one makes 4. This lavender block makes 3 different enzymes after it's cleaved. And these blue blocks are various kinds of accessory proteins that are needed, to assist the enzymes to pack the virus and so on. But this cleavage process is obviously going to be a pretty specific thing. The virus is going to be pretty finicky about where it cleaves the polyproteins. So there are proteases that are specific to HIV. HIV packages proteases in the virus. And that allowed people to say, if we could inhibit the HIV protease, maybe we can inhibit the HIV virus. And this is one of the early examples of success through what's called a rational drug design, where if you know the structure of the process that you're doing, you can, from first principles, design something to inhibit that process. And what these protease inhibitors share is that they have a tetrapeptide structure, kind of. That is, if you look along the backbone in here, it kind of looks like a tetrapeptide. This is the actual substrate in the HIV proteins with a phenylalanine hanging off there. And the various inhibitors have to keep something bulky there to satisfy that aspect of the enzyme. But then there's various kinds of tweaking that goes along around the edges for solubility and stuff like that. It's still not perfectly specific. Some of the HIV protease inhibitors have peculiar side effects with redistribution of body fat. We don't know what's going on with that. But it certainly is considerably more specific than just blasting it with, analogs to the, like methotrexate or something like that. So part of what's made HIV into a manageable, AIDS rather, into a manageable chronic disease is 2 different directions of inhibitors. You have the reverse transcriptase inhibitors and you have the HIV protease inhibitors. They're targeting it from 2 different directions. When I first began at Iowa, was at the very early start of AIDS as a phenomenon and an epidemic, and it was a mystery. People were just dying and they really didn't know why. And it's been in that time since then to now that they've been able to develop these inhibitors and turn AIDS from something that's a death sentence into something that's not much fun to live with, but is not as lethal as some other things that people can get. So let's take a little look at this. Here's a graph with 2 hypothetical enzymes, enzyme A and enzyme B. Which of these enzymes has the highest affinity for its substrate? Okay, why does A have the highest affinity? Okay, so in other words, if you look at its Vmax, which is somewhere around here, here's around half maximal. It takes maybe 2 micromoles to get it to half maximal, whereas you're looking at more like 5 or 6 for B. Which of these has the largest K-cat? B, okay. Why would you say that B has the largest K-cat? Right. Vmax and Kcat are directly related, right? They differ by the concentration of the substrate around. But so Vmax, so enzyme at the same amount of enzyme, B has a higher final velocity, so it has the largest Kcat. Which of them is probably allosteric? B? Anybody want to say why B would be allosteric? It's curved. It's curved how? Sigmoidal, right. So in other words, this simple curve is called the hyperbolic curve. This slow rise, picking up, tapering off, that's the sigmoidal curve. Sigmoidal curves are almost always allosteric. Which of these would be most active when you have 2 micromoles of substrate? Which one would be most active at 8 micromoles of substrate? B. So you couldn't say for these enzymes which one is most active. You'd have to always ask, well, how much substrate have you got around? Sometimes one's more active, sometimes the other one is. This is not pulled out of the air. There's a very important pair of enzymes that has more or less this relationship between their Vmax and Km, and we will come back to them because they're an exciting pair indeed. Okay. Let's see. I think I'm going to ask you to skip to the end because it's almost time is getting on. And we'll come back if we have time at that point. Let's skip down to 42. This is looking at an enzyme with the jawbreaker name aspartate transcarbamylase. And we're going to see that aspartate transcarbamylase is indeed an important and friendly enzyme. And we have here 3 different curves. We have this one in the middle that is marked no CTP or ATP. So CTP is cytosine triphosphate. ATP is adenosine triphosphate. And then we have one curve over here with CTP, and we have a different curve over here with ATP. How would you describe the effects of these 2 allosteric regulators on aspartate transcarbamylase? What is their overall effect? Someone want to take a stab at it? Anyone? Did you say increase the cooperativity? Why would you say it increases the cooperativity? So in other words, you're okay. I see what you're getting at is you're trying to sort of look at that low range and see if that's telling us something. But I think you've got a better handle on it there. That what the CTP has done is it's decreased the affinity. It has a lower affinity at this point. It has a larger Km. And ATP has the complementary effect of decreasing the Km, has increased the affinity. Affinity and Km are one of those pairs of words that tend to be kind of easy to scramble in your mind. Affinity is a sense of how tightly do you bind something, and Km is small if you have a high affinity because it doesn't take much to occupy it. And as it turns out, both ATP and CTP are part of the network of pathways that aspartate transcarbamylase is going to be involved in, so that it's getting feedback from a couple of different branches of the pathway. Let's take a look at this graph as well. This is a very complicated graph, and it's conveying the fact that enzyme regulation and allosteric regulation can have really complicated effects. I've used this metaphor for having a little molecular computer almost, that if you have a multimeric enzyme that's got allosteric sites tucked all over it as well as its active sites, that you can have several different compounds coming in and binding to it in order to modify the behavior of that enzyme. And on the left-hand side, we're looking again at this enzyme, phosphofructokinase. We looked at that before a little bit. Remember with phosphoenolpyruvate and ADP. This is now we're looking in mammals, not in bacteria. And in the left-hand graph, we're looking at the effect of something called fructose 2,6-bisphosphate. We're going to hear more about this when we get to glycolysis. But if you look here at the concentrations of fructose 6-phosphate that we're talking about, we're looking at between 1 and 5 micromolar fructose 6-phosphate. And we're adding to it 1/10 of a micromole of fructose 2,6-bisphosphate. Now, the product of this enzyme is fructose 1,6-bisphosphate. So this modifier is not binding at the active site. It's binding at an allosteric site of some kind. And which kinetic parameters is it affecting? There are only 2 kinetic parameters we've really talked about, right? It's affecting the Km very, very drastically. We can treat this as having essentially the same Vmax. So around here would be roughly 50%. And in the absence of fructose 2,6-bisphosphate, might take as much as 2 micromoles. And in the presence of it, it can pick up tiny, vanishingly small amounts of fructose 6-phosphate to push it forward. So this is a positive modulator. This is something that turns on the activity of this enzyme by increasing its affinity. It's also changing the shape of the curve, isn't it? That you see a somewhat sigmoidal shape here in the absence of it. That is, fructose 6-phosphate has a mildly allosteric effect on its own activity. But in the presence of fructose 2,6-bisphosphate, the activity curve is now basically hyperbolic. It's ignoring all other signals. It's just looking around to snaffle up fructose 6-phosphate. And we're going to see why when we get to glycolysis, why this is an important modulator. But this is a very powerful effect that it has. That's with respect to fructose 6-phosphate. So this is, again, this is a modulator, fructose 2,6-bisphosphate. This is the same modulator, fructose 2,6-bisphosphate, same concentrations as this graph over here. And we're looking at the enzyme activity again, but now we're looking with respect to a different substrate. We're looking with respect to ATP. You remember that this enzyme takes fructose 6-phosphate and ATP and is going to release fructose 2,6-bisphosphate, 1,6-bisphosphate, and ADP. So ATP is a substrate for this enzyme. And let's look at the activity of the enzyme in the absence of the modifier, this 2,6-bisphosphate. The enzyme activity rises and then falls. How can we think about that? Try to put it into English words. What do you think is happening there with ATP? Any thoughts at all? What this weirdness is? We're getting close to 420, so I'll give you an answer. ATP is definitely a substrate. It is also an allosteric regulator. So it's inhibiting the enzyme at really quite a low concentration. And one of the effects of this modifier here is to overcome the allosteric inhibition of the ATP. And now the enzyme can use the ATP as a substrate to make fructose 2,6-bisphosphate, a very weird enzyme. We're going to see it again. This isn't the only chance you're going to have to see it. Make sure that we get your cards if you want us to have them. Thank you. AV person? AV person? Yoo-hoo! AV person?
I did discuss a little bit some of the questions in the past and why we had to have them right for us. And I also made an announcement that I sent an email if we have a test for the 07. Students who had gotten below a 7 on test for the years to come and discuss youthful study strategies, and we would give up to 5 points for the students who did that, up to 7 for the test for some. When you come either during the regular office hours or if none of those before you could come and you can make an appointment for your second. You can meet with me or with Eli at that little one. Here's my magic AD person. I will have it as soon as the title. So I always say 4:30 to 5:30.